The Duncan Family Institute has funded me with seed funds in order to be able to use one of the biomarker assays that have been developed in my lab in order to use it for early detection of lung cancer and trying to identify people or smokers that are at high risk of development of lung cancer. And the way that we are doing this is using peripheral blood lymphocytes, or just a simple way of taking a sample of blood from the individual that we want to test, and do what we call a cytogenetic culture, in which basically we take blood and we put it in culture conditions which are mimicking your cells in the body, and we allow them to grow for 3 or 4 days, and then we harvest them, we make slides, and then we start looking at some of the cytogenetic abnormalities which are there. So overall, what the cytogenetic assays reflect is the overall genetic instability in the peripheral blood lymphocytes or in the individual's body. And genetic instability is well known to be a hallmark for development of cancer. We are collaborating with a research team at Rice University, which is headed by Dr. John McDevitt and his group. What we are doing with Rice University is that we are trying to automate our powerful assay, which we are having very much success with as a morphological assay, into a more automated form, which will allow it to be widely spread, heavily used in different large population studies. And basically what we are aiming at is that we can use this powerful assay for early detection of lung cancer. So instead of us having to look under the microscope and having to visually score 1,000 binucleated cells for each individual, what we are planning on doing with the Rice University collaborators is that develop what we are calling a lab on a chip. And basically it is taking our cultures, putting it on a platform, one of those very sensitive platforms that they have, and having the machine itself look at all the abnormalities that we would like it to detect. I would say that what we are doing is extremely novel. It has never been done before, and that's why we're very excited on moving it from just our regular culture-based, microscope-based assays into an automation that can serve a large population base. These funds that have been generously donated by the Duncan family are being used in translation research, which is basically taking our basic science researches that's being done in the lab and moving it towards a clinic in which we can apply it for either cancer risk assessment or early detection of cancer and cancer prevention, which really is going to be a strength in which we are identifying high-risk individuals and either putting them into programs which will allow this cancer prevention and giving them a cancer prevention edge, or early detection and therefore improve on their survival later on.